Hello. So this is Unit 4 of AS Bio, Cell Membranes and Transport, and this is the second section of the Learning Outcome Review, 4.2, Movement into and out of cells. Okay, the first thing you need to be able to do is describe and explain the processes of simple diffusion, facilitated diffusion, osmosis, active transport, endocytosis, and exocytosis. Simple diffusion is the net movement of molecules or ions from a region of higher concentration to a region of lower concentration down a concentration gradient as a result of random movements of particles. Oxygen and carbon dioxide are transported across the membrane via simple diffusion. And you're probably going to have to say that. Since simple diffusion occurs down a concentration gradient, it is a passive process, not an active one. So, I think we basically already said that, but when you're moving down a concentration gradient, you're going from an area of high concentration of solutes to low concentration of solutes. The way that my pre-ACE teacher taught us was like if you're climbing a ladder, if you're going down a ladder, it in this case doesn't require energy, but if you're going up a ladder, it does. So think of the concentration gradient like going up and down a ladder, but in this case we're talking about going down the concentration gradient. So it's a passive process, not an active one. The random movement of particles is caused by kinetic energy. So it is a passive process, but it still uses kinetic energy. If it were active, it would use ATP, but it's passive, so it uses kinetic energy. Energy is still required no matter what you're doing. It's just whether just random movement of particles causes that energy or whether your body has to produce that energy. I might have described that a little wrong, but the basic concept at its core. Simple diffusion involves the transport of uncharged and nonpolar molecules. Water is also transported across the membrane via simple transport. It can also be transported using aquaporins, but don't get aquaporins confused with channel proteins, which I'm about to get into. But aquaporins transport water. Channel proteins use water to transport. So remember that simple diffusion is a passive process using kinetic energy occurring down a concentration gradient. And there are a few factors affecting the rate of diffusion that you should probably know. The steeper the concentration gradient of a substance across the membrane, the faster the rate of diffusion of that membrane. Let me repeat that. The steeper the concentration gradient of a substance across a membrane, the faster the rate of diffusion of that substance. The steepness is the difference in solute concentration. So, these numbers make no sense. But if I have 20 solutes versus 2 solutes, that's one steep gradient. But if I have 6 solutes and 2 solutes, it's not that steep of a gradient. As temperature increases, so does kinetic energy, so the rate of diffusion increases. Large molecules require more energy to move than small molecules, so large molecules tend to diffuse more slowly than small molecules. As surface area increases, so does the rate of diffusion, because there's more area for the substances to diffuse through. Okay, facilitated diffusion. The diffusion of a substance through a transport protein, either a channel protein or carrier protein. Also, if on the test they say protein channel or protein carrier, it means the same thing, so don't let that confuse you. And a cell membrane. The protein provides hydrophilic areas that allow the molecular ion to pass through the membrane, which would otherwise be less permeable to it. Facilitated diffusion is still a passive process, so it does not require the use of ATP. Facilitated diffusion transports large polar molecules or ions down a concentration gradient. These substances are not lipid soluble, so they cannot diffuse across the membrane without a carrier protein or channel protein. Carrier proteins do not have a fixed shape. They flip between two shapes and are specific to a certain molecular ion. So essentially, like, it's in the membrane like this, and then the molecule comes in and it kind of just flips in the membrane, to my understanding, and that allows the molecular ion to enter the cell. So there's a lot more to it than that. There's that, like, the ATP interacting and it forms the ADP and the inorganic phosphate. There's a biofact sheet on the sodium potassium pump that explains that in depth. Carrier proteins, I already said that. Channel proteins are water-filled pores spanning the membrane. They have a gate that opens up and allows specific charged substances. It allows in a specific charged substance. They can also allow in molecules. So just hydrophilic molecules, polar molecules, that kind of thing. That's the same word. But remember that it's a water-filled pore. But it is not an aquaporin. Okay. Osmosis is the net diffusion of water molecules from a region of higher water potential to a region of lower water potential through a partially permeable membrane. Water potential is the tendency of water to enter or leave a cell. Water moves in the direction of more solutes. Water potential is negative, with 0 being pure distilled water. And that doesn't mean just like drinking water, like actually distilled in a factory water. Anything other than pure water has a negative water potential. If a substance has less solutes relative to a different substance, the water potential is less negative. So, which literally just means it's closer to 0 than the other water potential. So if there's more solutes, the water potential is going to be more negative. So water moves towards the substance with the more negative water potential. So if substance A has a water potential of negative 10 and substance B has a water potential of negative 20. This is in kilopascals, by the way. But substance A has a water potential of negative 10. Substance B has a water potential of negative 20. Water is going to move from substance A into substance B until they reach equilibrium. And equilibrium would just, at that point, isotonic solution, the net movement of water is 0. So when we're talking about hypertonic, hypotonic, isotonic, which I'm not going to touch on, you just need to remember that we're talking net movement of water, not just movement of water. Because even if the net movement of water is moving out of the cell, there's still water moving into the cell. And even if the net movement water is moving into the cell, there is still water moving out of the cell. Alright, active transport. The movement of molecules or ions through transport proteins across a cell membrane against their concentration gradient using energy from ATP. Active transport uses pumps to transport substances against their concentration gradient. Oh, that's all I wrote. I mean, it's active transport. It's an active process. Pumps are used. A good example is the sodium potassium exchange pump. So look into that if you wish. But really the main point with active transport is that it's against the concentration gradient and it uses ATP. Endocytosis is the bulk movement of liquids, penocytosis, or solids, phagocytosis, into a cell by the infolding of the cell surface membrane to form vesicles containing the substance. Endocytosis is an active process requiring ATP. Phagocytosis is cell eating, so the little particle will come up to the cell, and then the cell will form a vesicle around it and engulf it and eat it. And then pinocytosis is cell drinking. So it's similar, but they're smaller vesicles. It's the same process, just a smaller vesicle. It's important that when you think about this, you're not thinking about water, because again, water enters the cell through simple diffusion. So when we're talking about pinocytosis, more times than not, the cell is taking in lipid droplets. So in pinocytosis, the vesicle is forming around lipid droplets and taking that into the cell. It's not gonna be water, because that comes in through simple diffusion and aquaporins. Exocytosis, the bulk movement of liquids or solids out of a cell by the fusion of vesicles containing the substance with the cell surface membrane. Exocytosis is an active process requiring ATP. Exocytosis is just the reverse process of endocytosis. So, whereas in exocytosis, there's something on the outside, we're making a vesicle to bring it in. In exocytosis, there's a particle on the inside of the cell, and we're making a vesicle to push it out of the cell. And you can call these vesicles or vacuoles, by the way. Either one is fine. Unless, like, the question specifically says, use or vacuoles. I mean, it won't say it like that. It'll probably just say, like, in reference to formation of vesicles. Then say vesicles. Okay, number 2. Investigate simple diffusion and osmosis using plant tissue and non-living materials, including dialysis tubing and agar. So, dialysis tubing is Visking tubing. So, simple diffusion with Visking tubing. Visking tubing is a partially permeable, non-living membrane made from cellulose. Fill the length of the Visking tubing with a mixture of starch and glucose solutions. If the tubing is suspended in a test tube of water for a period of time, the presence of starch and glucose outside the tubing can be tested for at intervals to monitor whether diffusion out of the tubing has occurred. The results should indicate that glucose, not starch, diffuses out of the tubing. And then osmosis using plant tissues. Place pieces of beetroot into water at different temperatures or into different alcohol concentrations. Any damage to the cell membranes results in the red pigment leaking out of the cells by diffusion. Changes in color of the surrounding solution can be monitored qualitatively, visually, or quantitatively. Use of a color emitter. Three, illustrate the principle that surface area to volume ratios decrease with increasing size by calculating surface areas and volumes of simple 3D shapes. If the surface area is not much larger than the volume, the cell cannot take in substances at a fast enough rate to survive. A small surface area to volume ratio results in cell death. So, whereas these numbers are probably not even close to realistic at all either, but like if you have one cell where the surface area to volume ratio is 12:1, this cell is taking in so much nutrients, it's gonna be okay, it's gonna survive. Whereas you have another one and it's huge and your surface area to volume ratio is 2:1, and that cell's not going to survive because it can't take in essential nutrients quickly enough. Can't take in oxygen quickly enough. 4. Investigate the effect of changing surface area to volume ratio on diffusion using agar blocks of different sizes. Solid agar is prepared in suitable containers. The agar will be purple if it is made up of dilute sodium hydroxide solution and the universal indicator. Cubes of required dimensions are cut from the agar, placed in a container, and covered with different solutions, i.e. with a different solution, i.e. dilute hydrochloric acid. Convert to rates either the time taken for the acid to completely change the color of the indicator and the agar blocks, or the distance traveled into the block by the acid in a given time. The rate of diffusion can be plotted against the surface area to volume ratio. And your buzzwords for this unit are concentration or volume, but never say amount. And concentration and volume are involving different things, but you could say amount for either one of them, but don't, because they won't count your answer. So, say concentration when it's concentration, and say volume when it's volume, but never say amount. And then just remember to write out surface area to volume ratio in its entirety. And I did not talk about plasmolysis or turgidity at all. So, in a nutshell, when you have an animal cell and there's too much water going into the cell, a hypotonic solution, the cell is going to get real big and can burst. That's not good. And then you have an isotonic solution, which is just, the net movement of water is 0. There's the same going in as is going out. That cell's perfectly healthy. But then in a hypertonic solution, you have there's more water exiting the cell than going into the cell, and that causes, net movement, obviously. But that causes the cell to shrink. And both the bursting and the shrinking will lead to cell death. But that's with animal cells, I'm so sorry. That's with animal cells. With plant cells, it's different because plant cells have the cell wall. And so, obviously, there's the point where there's this perfect amount of water going into and out of the cell. But when there's too much water going into the cell, a pressure forms between the cell wall and the protoplast. And it's kind of just like this push back and forth between the two, and this is turgor pressure. And when it reaches the state of full turgidity, because of the cell wall, this cell isn't going to burst. And then plasmolysis, so there's too much water leaving the cell. You have your cell wall, you have your protoplast, but as there's so much water leaving the cell, the protoplast is shrinking away from the cell wall. And once the protoplast has completely detached from the cell wall, that's plasmolysis. And I do believe that cell is dead. And I think that's really it. Obviously, this isn't everything. And a lot of the things I say off memory. So if something I said sounds wrong, definitely double check it. I wasn't super strong with this unit, so I'm not saying that every word I said was wrong, but I probably made some mistakes. So if you heard something different elsewhere, don't take my word as gospel. But either way, I hope this was helpful, and I love you. You're going to do so good tomorrow.